bacteria are comparatively rare, and few outbreaks have been traced to infected milk handlers. These are the milk-borne diseases from human origin and bovine origin. Now we will discuss about the microbial examination of milk. So, how the milk gets contaminated, we have discussed. Now, the quantity of organisms or the amount of organisms present in the milk should be quantified. And for that, bacterial or microbial examination of milk is necessary. As the milk is used universally in the as a nutritive food, it must be free from microorganisms, at least the pathogens. Hence, it is necessary to test the milk for the presence of bacteria. The commonly used methods for enumeration of bacteria or examination of bacteria are the direct microscopic count (DMC), standard plate count or agar plate count, that is Specie, methylene blue reductase test or MBRT, and resazurin test, that is RT. The next is phosphatase test. We will discuss about the direct microscopic count. In direct microscopic count, the milk is the microorganisms in the milk is counted directly, and is the quantitative examination of milk. Normally, normal adults of the cow are probably never sterile. Organisms are present in abundance in freshly drawn milk. The first milk drawn shows a highest count, the middle milk shows a smaller count, and the strippings the least of all. The comparative high count of the four milk is the result of washing out of the easily removable organisms present in the milk process. The number washed out becomes less and less during the milking process. It is general practice to discard the first few streams of the milk which contains the high count. This may decrease the bacterial count by as much as 5%. The high bacterial count means that the milk has come from the diseased udders. It has collected under unsanitary conditions and has been kept warm enough to permit bacterial growth. It would seem, therefore, that the contamination of milk by bacteria is largely preventable. The method commonly employed for examination or microbial examinations are direct microscopic count, agar plate count, and methylene blue reduction test with resazurin test. The direct microscopic count we will discuss now. In this method, this method contains of spreading 0.01 ml of milk sample uniformly over the ruled area of 1 square centimeter on a glass slide, allowing a thin film to dry. Removing the fat, staining the film with methylene blue or crystal violet, and examining the sample under the oil immersion objective previously calibrated to determine the area of the field. The individual cells are the result expressed as number per milliliter of the milk. In this, a marked area of 100 millimeters or 1 square centimeter area is made with glass marking pen on the glass slide, spreading 0.01 ml or 10 microliters of milk sample homogeneously in the marked area, allowing the smear to air dry, removing fats and with the specific solvent, and staining the smear with methylene blue or any basic stain for 2 minutes. Counting the total number of bacterial cells within 10 different fields under oil immersion objective and calculating the average number of bacteria per field. calculating the area of microscopic field using the stage micrometer slide and number of microscopic fields accommodated within 100 micromillimeters per square millimeters area. Calculation of number of bacteria in 100 square millimeters, that is the sample 10 microliters or 0.01 ml. And finally, number of bacteria per ml of the milk sample are calculated. The calculations are made in the very much calculations are made in this procedure. The method possesses a number of advantages over the plate procedure or agar plate method. The result can be obtained quickly, usually at about 10 to 15 minutes. Since less work is required, more samples can be examined by this procedure than by the plate method. The amount of equipment necessary is much less. The slides may be preserved as a permanent record and examined whenever occasion arises, whereas plate must be examined and discarded. Some idea of the morphological type can be obtained from slide preparations. This is frequently of great value for determining the cause of bacterial count. Microscopic examination reveals the presence of leukocytes and other body cells in the milk. An excessive number of leukocytes indicate a diseased condition of the udders. The slide method gives a better quantities of determination than the agar plate method. Some believe that the method is less useful for pasteurized milk because most of the bacteria are dead. This is not necessarily so. It has been shown that bacteria killed by heat treatment usually disintegrate and gradually lose their ability to stain within few hours after pasteurization. Even if the bacteria remains intact, they are usually distinguished from living forms by their inability to stain intensely. The method is relatively quick. The results are obtained within 15 to 20 minutes. The equipments required are very simple and sterilization is not required. The slide can be preserved for more time. Morphology and arrangement of bacteria are observed, which may help in identification of species. Leukocytes and other body cells are also counted. These are the highlighting advantages of the direct microscopic count. The disadvantages are also there in this method. The method is being quantitative, do not differentiate pathogens or non-pathogens. The dead cells and sometimes particles are also wrongly counted, which is not significant. The individual cells in a cluster are difficult to count accurately. Homogeneous distribution of cell is not observed in different fields during counting. Some cells may remain unstained, such as acid-fast bacteria like Microbacterium tuberculosis. Therefore, the method is not applicable, and the method is also not applicable in low count samples or pasteurized milk. The basic difficulty is the differentiation or distribution, homogeneous distribution of the cells is not done if improperly the slide is not properly prepared. Then we will discuss about the Specie or agar plate method. This agar plate method is used more than any other method for enumeration of number of bacteria in the milk. The method consisting of preparing a series of dilutions of milk sample, transferring 1 ml of amount to petri dish, mixing with 10 to 12 ml of melted agar cooled to 45 degrees Celsius before pouring, incubating the plates at 37 degrees Celsius for 48 hours, and counting visible colonies developing on agar plate. The results are expressed as number of bacteria per ml per milliliter of undiluted milk by the standard plate count. The factor which influences plate count includes temperature of incubation, composition of plating medium, amount of oxygen, and these are the factors. Unless the standard procedure is followed, the result from different laboratories may show wide variation. The advantages of this method is that agar plate method is especially suited to determination where bacterial number are low. The method is generally used for testing pasteurized milk and a high-grade raw milk. It is the only method approved by American Association of Medical Milk Commission for examining samples of certified milk. The objection of this plate method is that it is the most numerous. The most important is the probably the pathogenic bacteria usually are not detected. If they grow, they cannot be distinguished from non-pathogenic forms by appearance. The number of distinguished forms from non-pathogenic forms by appearance. The number of colonies appearing on agar plate do not represent all organisms present in the milk. Many of the organisms fail to develop. Aerobic or anaerobic organisms do not find conditions favorable for growth. This means that no single medium or given set of conditions is capable of giving growth of all viable organisms likely to be found in the milk. The temperature, that is 37 degrees Celsius, is not the optimum for the growth of all organisms. Shaking the sample does not break up all the clumps or group of bacteria. Chain of staphylococci usually remains intact and recorded as only one colony. The colony count represents only a fraction of the total bacterial count in the milk. Agar count should be regarded as estimate rather than exact number because of the long incubation period. The milk is usually consumed before information of the number of bacteria present in the milk is available. However, the quality of milk supplied cannot be determined on the basis of one bacterial count. A series of count is necessary to establish the quality of the milk source. So these are the disadvantages of the plate method. The Specie or agar plate method using Specie, the total number of live bacteria in the milk and any source can be calculated. This method involves preparation of different dilution of milk samples, that is 10 to -1, 10 to -2, 10 to -3 using 9 ml serial dilution distilled water. Aseptic inoculation of 10 microliters of each milk dilution on separate nutrient agar plate by spread plate technique and incubation at 24 hours at room temperature. Counting the number of colonies in each plate and calculation of average number of bacteria per ml of the milk sample is done. The method counts the living organisms in the milk which are only metabolically active. Method do not determine the number but also does not only determine the number but also allows further study of biochemical character significant for identification of bacteria. Specific pathogenic species can be detected and enumerated using selective medium. This method is capable even in the milk sample having low bacterial population. The drawbacks of standard plate count or agar plate method is that it requires relatively more time, that is 24 hours. During this time, the milk sample would be already distributed and utilized in the community. The method requires sterile glassware and nutrient agar, which makes the method more complicated and increase the cost of analysis. Under the provided incubation condition and the medium, all types of bacteria cannot grow. Anaerobes cannot grow. Under aerobic condition and vice versa, anaerobes cannot grow. And under anaerobic condition, aerobes cannot grow. Incubation at room temperature allows the growth of only mesophilic bacteria, whereas thermophilic and sagrophilic bacteria cannot grow. So the bacterial count will be less. The leukocytes and other body cells are not counted. They are also important in determination of microbiological quality of the milk. So these are the two tests that are performed. The next test performed is methylene blue reduction test. Before that, we'll compare the Agar plate method or standard plate count with direct microscopic count method. In this comparison, the microscopic method gives much higher individual cell count than the Agar plate method. The difference between the count by two methods is considerably greater on samples showing low bacterial counts than those showing high counts. The organism in low count milk generally represent external contaminants, proof that fail to develop on agar, whereas those organisms with high count milk are forms which have developed in milk. Also, low count milk usually shows a greater percentage of clumps than high count milk. Bacterial standards are generally based on microscopic clump counts on agar or agar plate counts. The standards being the same for by either method. However, if individual cells are counted, the slide method gives higher result, the ratio being 3.33 is to 1. Therefore, total cell count by the slide method is approximately 3.33 times greater than that of the counts by bacterial plate count method. The word clumps refers to the isolated cell, whereas the group of bacteria, as well as the group of the bacteria. The next method is methylene blue reduction method. In this method, the method depends upon the ability of bacteria in the milk to grow and to consume the dissolved oxygen, which reduces the oxidation reduction potential in the medium. Certain enzymes present in bacteria known as oxidoreductases are then able to reduce oxidation by the removal of hydrogen. The methylene blue accepts the hydrogen and becomes reduced to the colorless compound. The speed of decoloration is an indication of the rate at which oxidation takes place. Methylene blue is of value in making a rapid survey of quality of raw milk. The rate of decolorization depends upon the number of organisms present. The test can be employed to determine in a rough way the bacterial population in the milk sample. The procedure is quickly and easily carried out and with minimum expenses. It is particularly valuable in making rapid inspections of large number of samples to determine whether the milk received by companies answers the requirement of the prescribed law. The test is expressed as the time required for the known concentration, that is about 1 is to 250,000 dilutions of methylene blue hyacinate to lose its color when the milk is incubated at 37 degrees Celsius to fasten oxygen consumption and to short the period of observation. Under some conditions, the blue color does not disappear uniformly. In such cases, the end point is taken as the time required for milk to show no blue color after it is mixed. There is not always good agreement between the methylene blue reduction time and agar plate count because some organisms fail to grow on the nutrient agar. The clump of organisms record as only one colony, whereas the rate of decolorization is due to the combined effect of each member of the mass. The rate of decolorization of the stain is not the same for all organisms, and the test becomes less accurate as the reduction time is increased. Freshly drawn milk requires at least 10 hours to decolorize methylene blue. Milk may be classified on the basis of methylene blue reduction